In progress.
One second here. Oh, that's where it is.
Recording.
Bear with me just for one moment. Like I had to do the other night, I'm just making sure that I have everybody who's attending the class on my attendance, because if you're not on there, you'll get dropped. So bear with me just for a moment here. I'm just seeing a name here that wasn't here the other night. No, maybe she was here. Bear with me really quick.
In progress.
Okay. I have it now. This is just a gentle reminder. The lab exercises are due the night before, bear with me here, Tuesday nights at midnight. So the ones that we did, so lab exercises 1 and 2 that we covered last week were due last night at midnight. If you did not do them, I need you to contact me and tell me when you think you're going to get them done. Like I've said before, I'm really lax when it comes to deadlines. I do have to give you deadlines, but I'm more than willing to extend them out if you have an idea as to when you're going to get it done. I would prefer that you don't get behind, and you will prefer you don't get behind, because it becomes a domino effect and you just get further behind. Okay. Christine, I understand. Any questions, concerns right now?
Well, question. Thank you, Professor. I'm on my husband's like 2 screen setup, so I can have like our online stuff on one screen and look at you in the other.
I understand.
But he works for the government, so on his home setup, he doesn't have a camera. That one question I emailed you about
You
lab stuff where we were labeling the different regions.
mm-hmm.
Do you have a question that's so picky like that on any of our tests or quizzes?
No, that's why I'm saying no. Just skip the question. I
Okay.
don't, that's a problem with Wiley. And I don't run into it very often with Wiley. I think I'm trying to think if there's ever, if I remember another question that came up last semester that was like that. And no, if there is, bring it up with me, but you know, especially on those, just skip it. I know that it's wrong. You know that it's wrong. I know that it's wrong. We can tell left from right. We all know it's wrong. Wiley is still better when it comes to lab exercises. But when you run into those questions, just skip them. I'm going to round up your scores anyway. And you're rarely going to have, actually, I think you have it tonight, but I don't think it's a bad question. You have less questions tonight. Some night we shall have more questions. Some you'll have less. This week you have less. And I think there's one that's worth a full point, but you also get partial credit on it. And so when you run into something like that, just skip it. And then if it really does dock your grade and bring it down significantly, bring it up with me then. Does that make sense? Because you won't run into them very often.
Yeah, thank you.
All right, someone told me they couldn't access it. If you tried to access it after midnight last night, you're not going to be able to access it. Why weren't you able to access it? What kind of message did it give you? I need to understand a little bit more about why.
It kept sending me to DTL homepage. When I click on it, it sent me right back to where I pick what class I want to click on.
Okay, when that happens, will you call Wiley and ask them? Because I think that was the same problem someone else in the class had.
Okay, is the numbers up there for me to contact Larry?
You know what? Did I give them to you the other night? I don't know. Did I give them to you? I thought I did, but let me give it to you right now.
Okay.
So the Wiley number, I'm going to give all three of them to you. Bear with me just for a second because I have to look them up on my phone. I call them less as I get better with their software. But because I've called as often as I have them on my contacts on my phone. So this is the Wiley IT tech line. And their phone number is 1-877-725-7503. The hack IT tech line. So if there's a problem with D2L or there's a problem with Zoom, it's usually that it's a hack IT tech who are going to be able to help you. Their phone number is, let's see if I've called them recently or not. I don't think I have, but I have them on my contacts. So give me 1 second here. Is 717-780-8255. 2570.
Okay.
And last one, McGraw-Hill. Tech is one 800-338-3987. So because most of you will even if you go into face-to-face classes, you will be submitting work online. First off, professors right now are very wary about picking up papers from students because of COVID. Almost everything is digital submissions right now. I personally am not really wary because I just wash my hands all the time. I have like a 5-year supply of hand sanitizer. But there are professors who are very wary about passing around papers. So you will be submitting things online.
So the person who had the same issue, did they get it fixed?
No, because they told them it would be 24 to 48 hours.
Oh, okay.
So I just, you know what, I need you, if this was Kathy, I need you to send me
Yes.
an email.
I did. You never responded.
When did you send it to me?
The day before yesterday.
Then you know what, I missed it. When that happens, do me a favor and text me, all right?
Okay.
Because I do get quite busy
Mhm.
and I will miss emails. I can tell you I'm flooded with them right now because it's the beginning of the semester. So do me a favor and text me because I did give you my phone number.
Okay. Yeah, I saw that, but
I'm
Yeah.
not gonna feel bad if you text me, I promise. You can harass me.
okay.
Okay, so these are the phone numbers, my phone number, but I can honestly tell you that if it's a computer issue, I am not going to most likely not going to be able to solve it. But my phone number is 952-201-3126. Are there any other issues or questions that have come up? Yeah, honestly, if it's a computer issue, I'm going to refer you to these numbers and these people because I honestly, I'm lucky I know as much as I do about my own computer. I can't solve other people's computer issues. My dad tends to be my computer go-to person and he lives in Virginia. So if I really need his help, I'll call him. But for some reason it skipped me in the gene pool to be that good with computers. Right. So tonight's lab, and you will see that as we move forward with the labs, some of our labs are going to be in parallel with what we're doing in lecture, and some of them are not. This is one that's not. It's a little ahead of some of the stuff we're doing in lecture. But ultimately, by the end of the semester, you're going to see that we're in parallel. But tonight's lab is over microscopy and cell biology. And so for this lab, you'll see you only have one Wiley lab exercise. And it's significantly smaller than the one you had last week. But you do have 2 virtual labs that are being assigned. And I thought they were opening today. And you know what? I'm going to change that right now. Because it shows here that they're opening at midnight tonight, and I don't want to do that. I want them to open before then. Bear with me. Actually, let's. You know what, Ruby, stop bouncing around the room, dog. I don't know if you can, if I mentioned it or not. I have two dogs here. I have this little one who's here snoring. So if you ever snore during class, she's the one doing it. And then, did I mention the other dog? Have I mentioned her so far? The other dog?
No.
Okay.
No, you mentioned your cat though.
Yeah.
I mentioned the cat. So the cat normally, because we now have a really big dog here. I live in kind of what's 2 apartments in 1 house, a duplex. Most duplexes are side by side. This one is a top and a bottom, and we live on the bottom. And our neighbor has been struggling with bladder cancer for quite a while. And he's been in a hospital, and now he's in a long-term care facility. And he has a dog who is 13 years old. And she's what they call, I'm trying to remember, Malinois. And for the last 1.5 months, we've been taking care of her. She's been living upstairs. But I can tell you that dogs don't like to be left alone. And so I can't get I'm just trying to get this to assign dates. Okay, it says, what's the problem here? Bear
I
with me
think
just
the
for a
time
second.
needs to be changed to
Current
like
date.
20.
All right, thank you. Let me change it. I'm going to change it to 7. We'll change it right here. Okay. She's been upstairs all by herself since he left, which was right around Thanksgiving. And all she does up there is cry and howl and bark. And my mom and I are both pretty big animal people. And I was mainly the instigator here, but I asked my mom to finally, let's just bring her downstairs. Because listening to her bark upstairs, first off, it's heart-wrenching for me. And she barks all night long. And so now she's here in our house And so if you feel my table bump and it jiggles every once in a while, she's walking underneath the table right now. She's keeping life entertaining. And her name is Ruby. I think she's looking for my mom, who's not here right now. Maybe. Okay.
While you're pulling up that information, I got a general question for everyone. For the individuals that live in the Lancaster area, where do you guys do grocery shopping? I can't seem to find a good place to grocery shop.
Well, I go multiple places. I do Walmart, Wise, and Giant.
I go to grocery outlet a lot and Aldi, really nice.
Okay, thanks for the recommendations. I just moved to the area, so I'm like trying to find my way around.
Aldi's is also good.
Where?
Aldi's.
Okay.
Recording in progress.
Grocery outlet is pretty cool because it has like just overstock from bigger stores. So you might find some of your like, I guess like more name brand things if you have specific items that you really like. And they have a pretty good selection. You just got to check for expiration dates, but things are super, super cheap there.
Wow. I know we have a grocery outlet. I live in Hanover, and I know we have a grocery outlet here too. But I like Aldi's and Walmart when I'm grocery shopping for the most part. So starting into this, at lab exercises 3 and 4, the first one is about microscopy. The second one is about cells and the structures within cells. So the first part is learning about the microscope, and bear with me one more shift. I may have mentioned this before, but I have a little tiny microscope. And I got it at a thrift store several years ago for about $5. And it's nice to have on hand for the most part. It's better looking at the ones my nieces and nephews get for Christmas. I realize this is actually a very nice microscope in comparison. But when you're holding a microscope, your typical microscope, if you get on eBay and start looking to buy a microscope, you're looking at several $1,000s for a typical microscope. Obviously, I lucked out with this one. But the reason I tell you how expensive they are is because we handle them with a lot of care. On this one, I'm not so worried about. The ones that you find in a typical lab, I worry more about. So the base is down here. Base is down here. The arm is here. The head is here. The light source is coming up from underneath and going up through your slide. In this one, because this is a cheap microscope, the light is actually coming from on top. But I told you it was a cheap microscope. I don't know who made it, but it's really quite cheap. The stage right here where you would put down your slide with stage clips to hold it in place. You have on big microscopes that aren't $5 at a thrift store. You have a coarse focus knob, and you have a fine that's usually a smaller knob within the center. You have two objectives. You have your ocular objective up here at the top, and you have your objective lens. So ocular lens, objective lens. Typically, all the ocular lenses have a magnification of 10x. The objective lenses, and let me actually back up a little bit. What does ocular mean? Do you remember that from like last week?
The eye
Eyeball.
area.
It means the eye. So this is why that's the ocular lenses, because that's the one you actually put your eye, you look in through via your eye. Your objective lenses are the ones you actually use to look at the object. So your ocular lens has a magnification of about 10x. Your objective lenses can range anywhere between 4x to 100x. Have you ever, and I know we've all done it at some point, have you ever looked through a magnifying glass? Something.
Yeah.
When you look through a magnifying glass, it is the same thing. That lens that you're looking through on the magnifying glass is the same thing as any of these lenses. The reason I mention that is, and I wish I had a magnifying glass right here. If you have one lens that has a certain strength, and then you're looking at an object like a bug or a hair or something, and then you put another magnifying glass right on top of it. What happens when you do that? When you take one magnifying glass, you got an object like right underneath here. You have one magnifying lens and you're looking through it, and then right on top of the one magnifying lens, you put another magnifying lens and look at your object. So now you're looking through two lenses. What happens?
Does the image flip?
It will flip. No, actually, I don't think it does. I don't think it flips. What else? But what else will happen?
Does it get smaller?
It
Just guesses. These are
it
just guesses.
actually gets bigger. It actually gets bigger. And
Yeah,
you know what? And go ahead.
doesn't it kind of like spread this? It spreads the like sample out a little bit.
It goes out a little bit. Yes. It enlarges in a major way because the strength of one lens is multiplied by the strength of the second lens. Does that make sense? Is that clear? That's the same thing with a light microscope. It's the same with any microscope. One lens is going to magnify the strength of the next. And it's just going to multiply it. And David is right about one thing that I was going to bring up later. He kind of jumped me on it. When we look underneath a microscope, the object is inverted. I don't think you see it with a magnifying glass. I'm pretty sure you don't. But you do see it with the microscopes. So your left becomes your right, your up becomes your down. And with a little bit of practice and using a microscope, you get used to that. So looking at here. Looking at this. Bear with me just for a second here. So looking at this, here's our light source. It's shining light up through this hole in the stage where our slide will be with the object on it. And then we have our ocular lens and our objective lens. When we want to measure the amount of magnification of any object we're looking at, we multiply the strength of this lens by the strength of this lens. So if this is 10 and this is 10, we have 100x magnification. If this is 10 and this is 4, we have 40. One of the most, and probably the most expensive part of the microscope are the lenses. If you damage a lens, the microscope is useless. So we tend to be really, really protective about the lenses. And so normally when we go to rotate, go from one objective to the next, we only move them using this revolver up here. Because we don't want to damage our lenses. The reason that my microscopes are so expensive is because of the precision of the lenses. So you have a coarse focus knob, you have a fine focus knob. When someone first puts a slide underneath the microscope, they only use they use their lowest ocular objective lens, and they use only the coarse focus knob to bring things into focus. I usually, and I've learned the easiest way to do it is to take the stage all the way down, lower it all the way down using the coarse focus knob. So with this one, I don't have anything. I have a slide on here, but it doesn't have anything on it except for dust. But what I do is I bring it all the way down. And then watching it, and I don't wear my glasses because my glasses, I feel like they just get in the way. Let's see here. I work to bring whatever I want into focus by bringing up the stage gradually. And so I start on the lowest magnification and start bringing it in to focus gradually until I've reached until I've got it in focus. And if this microscope had a fine focus, the moment that I change objective lenses, I no longer use that rough focus. I only use the fine focus at that point. And the reason for that is because if I only use the fine focus, I will not run my objective lens into my slide, breaking my slide and my lens. And over time, the more you do it, the easier it gets. So the ocular lens, the objective lens, the revolving nose piece, the stage where you're going to put your slide, the stage clips that hold your slide in place, the condenser, which will actually condense the light rays that are coming up from your light source. You have a light intensity knob and the coarse and fine focus knobs. Any questions specifically about looking under using a microscope? I know that's a lot of information when you actually don't have the microscope in your hands. This iris diaphragm works like the iris of your eye, changing the amount of light that's moving through the specimen. That's why they call it your iris diaphragm, is because it's working like the iris of your eye and changing the size of your pupil or changing just how much light is going in. So some basic microscope vocabulary. Monocular, if you think ocular means eye, monocular meaning you have one eyepiece, like the one that I have. Binocular meaning you have two eyepieces. I don't use this microscope. It's mainly for show because I'm kind of a geek. But I actually looked at buying a microscope last semester, and that's when I realized just how much it was going to be. But I would prefer a binocular one because I like using both of my eyes. Your scanning objective lens is your objective lens with the lowest magnification. Then you have a low power and a high power. Usually you have about 3 objective lenses. So you have the scanning, you have the low power, and you have a high power. Most and almost all microscopes, another reason for the expense of microscopes is that if you get one thing into focus using your lowest magnification, and then you shift up to your next magnification, your image is almost completely in focus still. That's what parfocal means. Is that moving from one magnification to the next magnification is not going to change how clearly the contrast is in what you're looking at. You may have to make some really small adjustments with your fine focus knob, but overall you see the same thing. And to give you a better definition, or sometimes maybe just being able to see it, bear with me just for a second here. Parfocal. Oftentimes we call light microscopes compound microscopes because one lens compounds the strength of the second lens. Bear with me, that wasn't what I meant to do. So they interchange without varying the focus. So you can change out lenses without varying the focus. Magnification is how big can you enlarge what you're looking at? What's the difference between magnification and resolution?
Would resolution be how detailed and clear something is?
Pretty much. How much detail can you actually see? You can enlarge things really, really easily. I can make things really, 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 really big. But if I can't distinguish one thing from another because it's all a smear, that's my resolution. So for example, I wear glasses, take off my glasses, and I'm not really blind, but I'm blind enough that I have to wear them to drive. And I'm blind enough that I can read my screen fairly well with this enlarged print on the PowerPoint, but my glasses make it a lot easier. So I have my finger out about as far, and I can see it fairly well. As I keep bringing it in, it becomes bigger and bigger and bigger, but the edges become blurry. And I can keep making it bigger and bigger and bigger. But when I can't distinguish anything on it, that's the resolution. So resolution is distinguishing one point of something from another point. And how well can you see 2 points or 2 lines? So one thing is we'll talk about it as we go forward here. Microscopes are really great at magnifying things. But with technology, the resolution of microscopes has improved drastically. So a lot of times when you're looking at expense and technology of microscopes, you're looking at resolution. Is how much better resolution does one give you over another? The field of view is what do you see underneath the microscope? When you're looking at the specimen, how much of the specimen do you see? Now I want to stop just for a second here, and we're going to watch a video here that I found. Giving maybe even a better description of light microscopy.
My name is Ariel, and I just started my first job at
Maybe.
He knows a lot about the science stuff, Professor Dave explains. In the previous tutorial, we outlined some details regarding the history of microscopy and the main types that were innovated in the 20th century, which we commonly use today. Out of all the different and fascinating looking microscopes that scientists use to study microorganisms, the most common one that anyone will use in their early microbiologist ventures is the light or optic microscope. This is also the type of microscope that
What does optic mean? It means eye.
just about everyone is familiar with and would recognize instantly from high school biology class. But there is a lot to talk about in terms of its operation.
So monocular meaning what they're looking at right here. Binocular meaning what these are. You can see there's two eyepieces.
High school biology class, but there is a lot to talk about in terms of its operation. So let's get a closer look now. The light microscope, as the name implies, uses light to visualize something. The light passes through a lens, which focuses the light onto the specimen. After passing through the specimen, the light passes through the objective lens, which magnifies the image from 4 to 100 times its original size. Next, the light travels through the tube of the microscope and the eyepiece, which further magnifies the image by a factor of 10 or 15 before it finally enters your eye. Some light microscopes can magnify an image up to 1,200 times its original size. In order to use a microscope properly, one must know the names of all its components, as well as their functions. So let's take a look at a typical light microscope. Any light microscope has a stage that the specimen sits on, and stage clips to hold the specimen in place, which is usually nestled between a slide and a cover slip. Attached to the stage is the diaphragm, which controls the contrast of the image by controlling the amount of light from the light source that you expose the specimen to. On the sides of the stage are the slide adjustment knobs, which are used to move the specimen so that what you want to see is in your field of view. Above the stage are the objectives that are held by the nosepiece, which can be rotated so that different objective magnifications may be employed to view the specimen. The tube of the microscope connects the nosepiece to the eyepiece, which further magnifies the image and is the part one looks through to view the specimen. There are also focus knobs on the side of the microscope, the larger one being the coarse focus knob, used for larger changes in focus, and the smaller one being the fine focus knob, used for smaller adjustments in focus. To put these parts to use, one can start by plugging the light microscope in and turning on the light source. Then secure the specimen on the stage using the stage clips before rotating the nose piece so that the smallest objective is in place over the specimen. First, use the coarse focus knob to clear up the image. Then use the slide control knobs to center the specimen in your field of view. A tip for this is that the image in the microscope is inverted, so when you move the slide to the right, it will move to the left in your field of view, and vice versa.
So this is what David was talking about, that it's inverted.
Next, use the fine adjustment knob to refine the image. This can also involve adjusting the eyepiece or eyepieces to further focus the image of the specimen. A tip for adjusting eyepieces is that if there are two eyepieces, the focus for either eye might be different, given that your eyes are not identical. Additionally, the focus setup differs slightly between individuals with different prescriptions. Each person may have to readjust the focus when using the microscope. After the focus is set, rotate the nose piece to the objective appropriate for your purposes. Usually, between switching objects, it is necessary to use the fine focus knob to clear up the image, but avoid using the coarse focus. Make sure to note what objective you are on when you make observations. Also note that the total magnification will be 10 times the objective, as most eyepieces magnify the image further by a power of 10. It's important that when using the 100 times objective, it is set up for oil immersion to improve the resolution of the image. To do this, rotate the nose piece so that the specimen is between the...
So an oil will actually give you higher resolution when, remember, if it's at 100x here and your ocular lens is 10x, that's 1000x magnification. And if you remember, I made the comment that you can keep magnifying things, but if it's just blurry, you're not really seeing things. It's just getting bigger. And so the oil can actually improve the resolution when it's at when the compound microscope is at its highest magnification.
The 40 and 100 times objectives, and add a drop of oil on top of the cover slip, which sits over the specimen. Then rotate the nose piece so that the 100 times objective is submerged in the oil and over the specimen without scraping the lens. Finally, after making all of your observations with the light microscope, you will need to turn off the light source, unplug the microscope, dispose or save the specimen properly, use lens paper to wipe off any oil on the lenses, and replace the microscope cover.
And you only specifically use lens paper. You don't use like a paper towel, you don't use a tissue, because you can scratch the lenses. And like I said before, they tend to be the most expensive parts. So we tend to be really cautious about how we take care of them.
Light microscopes are commonly used to study bacteria, slices of tissue, liquid samples, and some mitochondria. Inverted light microscopes, which have the light source coming from the top and the objective under the stage, are often used to study tissue cultures, like cells covered by liquid media, as cells settle to the bottom of the flask, where the objective can pick up the image. This is in contrast with a standard light microscope, with the light source below and objective above the specimen, which would have trouble visualizing the cells through the liquid medium. Light microscopes are easy to use and also relatively inexpensive, making them common in high school and undergraduate labs. Also, with a light microscope, one can visualize living specimens over time with a low likelihood of damaging the specimen. However, this microscopy technique is limited in resolution and therefore magnification, making it impossible to see structures in as much detail as with other microscopes that we will talk about in a moment. So let's move forward and check out something much more powerful. Thanks for watching, guys. Subscribe to my channel.
So like he just said, light microscopes are great in a lot of ways, but they are limited in their magnification resolution. And so technology has worked to improve microscopes. And actually, before we do that, let's talk about a little bit more here. Wet mounts. This is probably one of the easiest types of slides to prepare. And what's done here is you take a little bit of a cheek, you kind of scrape the inside of your cheek, and you take off what are called, oh, bear with me here, epithelial cells. And starting next week, we will talk about the 4 basic types of tissue. But the first one is epithelial tissue. And so on the inside surface of your mouth is what we call non-keratinized epithelial tissue. And it's multi-layered, which in this with regards to epithelial tissue, we call non-stratified, or excuse me, stratified, non-keratinized stratified epithelial tissue. And it's squamous because the cells are flat on top. The reason I'm bringing this all up right now is because this is one of the easiest things to look at underneath a light microscope. And so what you do is you gently scrape the inside of the cheek and take off just the top layer. Just take off a little bit of the top layer. And then you add a little bit of dye. Why do you think you have to add dye? Why would you add dye?
It gives a better contrast of what's on the slide.
Why, you're right. Why do you think you need to add contrast to it?
Because otherwise it's all kind of translucent.
It's all translucent. Yes, it's all clear. The dye actually adheres to the structures and allows you to see them. The center here are the nuclei of the cells. So like, why was the stand, and what structures do you observe here? Really, all you're really able to observe here are the nuclei. We're going to stop again. I have another video showing exactly how this is prepared, so bear with me here. Can I get there?
People with ADHD save 10 hours studying.
In this video, I'm going to show you how to prepare a wet mount slide of eukaryotic animal cells, specifically epithelial cells from the inside of the cheek. Hello. Let's put on our eye protection to start. Here are all the materials that I'll need to wet mount my specimen, my microscope slide. And instead of using water, I'm going to be using a 0.9% sodium chloride solution.
We will talk about why she's using this 0.9 sodium chloride solution, but let me ask first, do we have anybody here who works in a hospital right now? Or works in a pharmacy right now? I think I heard something, but it wasn't really clear.
Oh, can you hear me? It's Christina.
I can. Christina, do you work where do you work?
I work in an emergency department at Hershey Medical.
Okay. Let me ask you a question. Why, when they go to give someone an IV, why do they make it 0.9% sodium chloride?
Yeah, actually, I'm one of the patient care secretaries, so I don't do as much.
I understand. No, I understand.
I can answer that.
Go ahead, say that. Go ahead, Zena.
Can you hear me?
Yep.
Because it's half sodium chloride, so it's half salt, half water.
Mm-hmm.
And
why is it 0.9 and not like 0.5 or 0.1?
Wait.
Do you need like an uneven mixture? Like, do you need more of one than the other?
Say it again. Say it a little louder.
So it sounds like you need more of one compound than the other.
Right. The reason they use this is because the ions, the amount of molecules that are in sodium chloride at a 0.9% is the same as what's within the cells.
The water.
Because
Yes.
once you lose fluid, you lose some salt. So to balance it out, they use the chloride.
Kind of. They need both of them, and we'll talk more about this in a couple of weeks. But they're specifically doing it so that the cells don't shrink or explode. And it has to do with what's called osmosis. If you want to jump ahead, it's OSMOSIS. And what it means is that if the molecules aren't the same on the outside of the cell and on the inside of the cell, when you put them in the cells in that solution, the cells will either shrink or explode because the water moves in or out. It shrink the water if there's too many ions outside, the water will move out of the cells and the cells will shrink. If there's too little molecules inside, now that you put it in this solution, the water will move into the cell and the cell will explode. So this 0.9% is the same as what your body has of sodium chloride ions. This is why when people need to receive IV fluids because they're severely dehydrated, they give them this 0.9%, is because they don't want to destroy your cells or put you into severe dehydration or water toxicity.
So did you say if there is like more salt in the intracellular and more water on the outside, then like the water be pulled to the cells? But if there is,
say it one more time. I'm sorry. Say it one more time because I'm hearing impaired. Say it one more time.
Okay. So if there is more salt inside in the intracellular, but there is less water on the outside, then they will pull the water inside because there is more salt in the intracellular.
Exactly.
Okay.
It has to do with, and we will talk a lot about this in a couple weeks. It has a lot to do with the fact that water moves from a high, everything moves from a high to a low. Everything is always moving from a high to a low. And the best example that I can give you to explain this high to low idea is, let's imagine that we're all in a room, okay? We're in a room. And rather than just having 20 people here, so we have 200 people. And this room is connected to another room. And there's a door right here, maybe another door right here. And we're in this room, and there's 200 of us in here. Actually, that's terrible. I will tell you that trying to write on a Zoom whiteboard is like learning to write all over again. So bear with me here, 200 in here. And there are 4 people in this other room. Okay. 4 people in this other room. This room, we're going to say it's 10 by 10. So it's about 100 square feet of space. And there's 200 of us in here. This other room is also 10 by 10, but there's only 4 people in that room. We're all in here. How many of you are going to try to get out? Yeah, David, I agree with you. I'm out. Samantha says she's out too. It's just because nobody wants to be in that high of a pressure room. It's the same thing for the water. It's the same thing for any molecule. We all, keep this in mind because I told you I teach 122 and I'm teaching it at the same time right now. A lot of physiology is that things move from a high to a low all the time, unless you physically force them not to. If you look at if you put dye in water, does it all stay when you drop that dye into the water? Does it just stay in the center? No, it moves out until it's all equally balanced and everything has equal amount of space. That's the reason she's putting these cells into this 0.9% sodium chloride. And I'm sorry for going off on my tangent. Let's come back to this.
Solution is isotonic compared to the fluid inside the cell.
Isotonic, meaning it's the same amount of molecules in this solution as it is in the cells. And that's why the cell doesn't explode or shrink.
Cells. If I used water, the cells would burst from osmotic pressure. Try to place a very small drop of the sodium chloride solution on the slide because we'll be adding more fluid when we add the stain. I'm going to use a toothpick to obtain my specimen. I can get a good sample of epithelial cells simply by running that toothpick gently along the inside of my cheek. I place my sample on the slide simply by swirling the end of the toothpick around in the drop of sodium chloride solution. The used toothpick is a biohazard and must be disposed of properly. The stain I'm going to use is called methylene blue. Cheek cells, epithelial cells are very transparent, and in order to see them under the scope, I'm going to need to stain them. Now that my sample is prepared, I need to put a cover slip over it, this clear square. The best way to apply the cover slip is to hold it at a right angle to the slide and then let it drop. This helps avoid the formation of air bubbles. Sometimes if you use too much liquid in your sample, when you drop that cover slip, the liquid can seep out the sides. I like to keep my slide nice and neat, so I take a paper towel and blot up any excess. Remember that sample has cells in it, so the paper towel will need to be treated as biohazard and disposed of properly. This is what my slide looks like when it's finished. Now I'm going to show you what these cheek cells look like when viewed through a compound light microscope. Here are my cheek cells at 100.
So let me say something. She says that 100x TM, which means total magnification. What do you think her objective lenses are? What do you think the magnification is? What's the magnification of her ocular lens? What's the magnification of her objective lens?
I would
10.
think that they're both 10.
It would be approximately 10 and 10.
Total magnification. You see they look like little fried eggs, and the blue dot inside them is the nucleus. They stick and clump together because they're sticky. They're cells of the mucous membrane.
What about here? When they're 400, what do you think the magnifications are? You don't, the ocular lens doesn't change.
$40 times.
So the objective lens would be what? What do you think, Sherry? 40. And the ocular lens is 10. When he was talking about adjusting the magnification for your different eyes, I don't really know what he was talking about in that video, because I can tell you that my eyes have 2 different prescriptions, but I don't think there's a way to do that. You can't alter the ocular lenses, but you still will get so that you can see it in focus.
I think he must have meant that the resolution, because I've had it where like people with different, different prescriptions have had to, mess around with the fine knob after I found it to be,
Right
clear
where
and
you
stuff.
want it. Right.
Yeah, and they'll have to change it for their own eyes.
Right.
But it didn't involve a magnification change.
Yes, I don't, when I'm using a microscope, like I said before, I don't wear my glasses. And so when somebody else tries to use it, they have to adjust it.
Magnified 400 times their actual size.
So what is the benefit of that oil immersion? Do you remember that from before? Why would she do oil immersion? Why doesn't she just use 1000x?
Resolution.
It's resolution. Justin's right. It's all about resolution here. You can keep enlarging something, but if it just gets blurry, that's not doing you any favors.
Here's a cheek cell viewed under oil immersion. That's at 1000 times its actual size. And you can see bacteria that's come from the mouth on and around that cheek cell, the little dots that you see in clumps on the cell and around the cell. Those are oral bacteria. More seriously,
I can say that I've never watched that. I didn't watch that video all the way through, but I like that because when you're in a typical A&P biology lab, you won't go into that 1000x oil immersion for cheek cells. And I didn't ever think about the fact that you'd start seeing bacteria in there too, even though you have a lot of bacteria in your mouth.
Does it matter what kind of oil you use for those slides?
I'm trying to think what they use. I don't. You know what? You're asking a really good question. And the fun thing about this class being online and that we have really easy online access is what oil is used for oil immersion. And one thing I'll tell you right now, in your typical college class, they don't do oil immersion because people are too good at breaking the lenses when they're doing this. And so they actually, at Carroll and Stevenson, where I teach also face-to-face, they take these lenses off so that students can't use them and break them.
Yeah, I took a AMP class in Central Penn, and when we used the microscopes to look at like urinalysis and stuff, we didn't have oil immersion. Like we had the blue dye, but we didn't have the oil immersion.
Right, you know what, you just don't use it. And it's partly because they're easily broken. It's a silicone oil. So silicone immersion oil is used for these deep observations. And let's see if there's any other kind of oil that they use. I don't see it. So silicone oil. Coming back. I love it how it always takes me back to the beginning. Not really. So a compound light microscope, like I said at the beginning, it's a set of lenses where the one magnification of one lens is going to be multiplied by the magnification of the next lens. And it allows you to focus the light rays on your eye and moving into the specimen. A transmission electron, the major difference when you're going from a light microscope into electron microscopy, and this is the simplest way that I know how to explain it, is that rather than using light waves, you're using waves of electrons, or you're using beams of electrons. That's the only difference. Is rather than using light waves that have a wavelength that moves, you're using beams of electrons that also move at a motion, a rhythm. And rather than using just glass lenses to focus those beams of electrons, you're using electromagnetic fields. So you're creating a magnetic field to focus them. Because as we'll talk about as we get into the next chapter of our textbook, Electrons are negatively charged. And what this does is it uses them to, and they bounce off your specimen. They refract off your specimen just like light does. If you look here, when you're looking at this picture, and I'm going to zoom in on it for a second. Looking at this picture, so this is 200x magnification. And you can see a fairly good amount.
I don't, all I see is the lab exercises 3 and 4. I don't see the picture that you're showing.
You don't see it. Let's try this again. Do you see it now?
There we go.
Okay, I'm sorry.
That's okay.
Do me a favor and tell me that every time. Oh, no. This is not going to work very well. Come here. Here. So looking at this picture here, and I will tell you that David was right about the fact that cells tend to be translucent. However, if you look at a plant cell, the green, the chlorophyll, is not translucent. It's the only thing that you don't need a dye for. You'll see it actually fairly well because it's green. But these are euglenia, which are single-celled animal that is capable of photosynthesis. And so looking at it, you can see a couple little things in there, right? You can see little tiny things in there. However, when you increase the resolution using electron microscopy, do you see the difference in what you see? Yeah. And the other thing about this is, and I'm gonna zoom back out again. The other thing about this was 200x magnification. This is 12,500. If you do this without increasing the resolution, it just becomes a blurry mess. You don't see anything. But the electrons, the beams of electrons are improving the resolution. And when we say transmission, this is called a transmission electron microscopy, because the electrons are moving through the specimen, just like light waves are. Remember that for one moment. The electrons here are transmitting through the specimen. So we're using two electromagnetic lenses that are creating these magnetic fields to focus the beams of electrons onto our specimen. And just like light, when it hits our specimen with the compound light microscope, the electrons are being refracted off because they move in waves also. They're being refracted off, and their refraction pattern is what generates the picture. So moving forward, now, rather than a transmission electron microscope, now we're using a scanning. What's the difference here? Here's this cell where you're seeing all the internal parts. What's this? What are you looking at here that you didn't see? These are actually, I didn't think about this, but this is the same as this.
You're looking at it in more of a 3-dimension.
You are. And are you being able to actually see through it?
No.
No, you're not seeing through it at all. Scanning electron microscopy allows you to look at surfaces and 3D surfaces. And what they do is they fix the specimen and then they coat it with a thin layer of metal. And the electrons, rather than hitting the specimen and going through the specimen, are bouncing off the metal. And you're looking at a refraction pattern of the way the electrons bounce off the metal surface that's coating this specimen. Are there any questions? Microscopy is really, really fascinating. When it's been pretty amazing how much we're able to see. And as we resolve things, you can see that a lot of your little bacteria that cause such havoc in your body sometimes have actually pretty cute little faces when you start looking at them.
Professor Gessel, where in the lab manual would we find out more of this? Or are we just supposed to take this information from the
I'm trying to remember in the lab manual, bear with me just for a second. Let me look at my one. I think the lab exercise 3 is just referring to the compound light microscope. The reason I'm showing you the others is because we will see them. We're gonna see pictures, and it'll make your life easier if you know how it's done and what specifically we're looking at. But your lab manual in lab exercise 3 is specifically talking about compound light microscopy and the wet mount cheek cells.
I have a quick question.
Go ahead.
In these electron microscopes, is there a way to like see the image that you're trying to get, or is it just kind of luck?
I honestly, I think it works the same way as the compound does once you learn. I have used high resolution once. I've used fluorescent once. I've never done this kind of microscopy. I'm lucky I understand it well enough to explain it reasonably well. I think if you got online and you wanted to look at YouTube videos asking how do they do this, you could find videos. So knowing the structures and their basic functions, there are a lot of organelles in a cell. If you remember, we talked about that hierarchy of organization, and that we start with atoms, then we move into molecules. Molecules become macromolecules. Macromolecules become organelles. Organelles become cells. This is specifically looking at the organelles. within a cell. This lab is going to go through them fairly quickly. And in 2-3 weeks, we will go through chapter 3, which is all about looking at all the cells and all the organelles and their functions. But for tonight, I've given some summaries, bear with me here, of each of what their functions are. But for right now, this is a review. This is a review and showing all the organelles that we could possibly talk about. So the nucleus tends to be the biggest, which is where the DNA is contained. The nucleolus contains a lot of RNA and ribosome proteins, which we'll talk about later. You have smooth endoplasmic reticulum where fats are synthesized. We have a rough endoplasmic reticulum where proteins are synthesized. We have ribosomes that work to synthesize proteins. And if you're not sure what a protein is, wait, because we'll be getting to it in the next couple of chapters in lecture. Just understand that they synthesize proteins. The plasma membrane is the most outer edge, and it is essentially the boundary between what's inside the cell and outside of the cell, and regulating what goes through, what is allowed into the cell and what exits the cell. The mitochondria is going to generate the energy for the cell. The majority of energy that's produced within the cell is going to be produced in the mitochondria. So this is essentially your powerhouse. It's in our houses would be generating the electricity. Lysosomes. Did I mention this before? What lysis was? I will tell you that if you say that I haven't, just feel free to tell me that, because like I've said before, I teach 6 classes right now, and a lot of them are at different levels, and I can't remember sometimes what I've said in one class and what I haven't said. So lysis, anytime you hear the word lysis on the end of a word, it means to destroy something or break it up, break up. When go ahead.
I'm sorry, are they part of that macrophage or whatever it is called?
They are. They're really important there. Zena, you're jumping ahead here. The reason she knows this is because she's also taking 122 at the same time. Zena, you're going to kill me in here. You know that, right? Because I'm like, wait, how does she know? I know we haven't talked about that in here. All right. All right.
I was just putting 2 and 2 together. I don't think we talked about that either.
We haven't, but you're right. You're right. So lysis meaning to break up. And no, I don't recommend you take 121 and 122 together. We'll just say Zena's a little nuts, because I wouldn't do it. Do it the way you're doing it right now. Don't do what Zena's doing. So lysis meaning to break up. So a lysosome is where things get broken up and broken down. So this is essentially your stomach or your vacuum or your place where things get destroyed. This is the garbage plant. things get put in here so that they get broken down. Centrioles work in generating the roads that are needed for things to move from one place in a cell to another place. So they're what generate roads and generate paths. The centrosome matrix is essentially just the surrounding of the centrioles. Cytoskeleton is what forms the structures of a cell that'll give the cell a particular shape, the overall cell, a particular shape. It works just like your skeleton. Some of us have bigger bone skeletons, some of us have littler bone skeletons. If we removed all the skin from our faces, we'd see that our skull takes on a very similar shape to our face shape. The cytoskeleton is providing the shape. We'll look, and as long as Zena doesn't overjump the gun again here, we'll look at how cells will move based on changing these cytoskeletal elements. And they can move, and like Zena said, they actually, what she was referring to is macrophages, or cells that engulf back other things, engulf other cells, and we'll talk about this, Zena, tomorrow or Friday. They engulf other things and break them down. They're part of your immune system. But we need these cytoskeletal elements to actually allow cells to take things into themselves to break them down. So it's kind of like your bone, your cytoskeletal elements are kind of like generating bones to allow you to pick something up. And your bones are your cytoskeletal elements. In this case, rather than calling them bones, we have microtubules and intermediate filaments. We have one more that's not here. A peroxisome is similar to a lysosome, except it's also involved in breaking things down, but it uses hydrogen peroxide. That's where it gets its name. It uses hydrogen peroxide. Secretion being something being released. Vesicles here are transported, they fuse with this plasma membrane and allow things to be released. I'm going to introduce some other keywords here. So cyto means cell. Exo means exit. So exocytosis is when something exits the cell. On this same line of thinking, endo is when something enters a cell or And endo means to enter. Endocytosis means to enter the cell. So throughout this semester, I'm going to spend a lot of time going through these root words and base words with you, because you will get better and better at looking at big words, because physiology, anatomy, and biology is all about really, really big words. If you can break them down into individual parts, you can take a really good guess what they mean. So secretion via exocytosis. So something is being released from the cell. Golgi apparatus is involved in, it's the mail postal office. It's involved in packaging things and putting stamps on them and determining where they're supposed to go next. Are they supposed to leave the cell, or is a substance that's been produced, is it supposed to go to a particular organelle? That's what the Golgi apparatus does. The ribosomes are involved in protein synthesis. The rough endoplasmic reticulums are also involved in protein synthesis. And we'll talk more about that in when we talk about chapter 3 in lecture. Any questions about this picture? So the plasma membrane is kind of a big deal because it separates the outside of the cell, the extracellular area, from the intracellular, the inside of the cell. It regulates what's going to come in and out of the cell and when. A couple different structures here. Glycolipid. Glyco means a sugar or a carbohydrate. Lipid is an oil. And the other one that we saw there a minute ago was protein. So we have these glycolipids. So glycolipids meaning it's a sugar or a carbohydrate attached to a lipid. The plasma membrane is made of what's called phospholipids, which is a phosphate attached to a lipid. Phosphate is a particular molecule, which we'll talk about in chapter 2. Just know for right now that a glycolipid means that it's a lipid attached to a carbohydrate. A glycoprotein is a carbohydrate or sugar attached to a protein. We have this phospholipid bilayer. What does bi mean?
Two.
It means two. So what that means is that there's 2 layers of phospholipids. Within this membrane, there's also cholesterol, because cholesterol is really good at altering the fluidity of this membrane. This membrane, it actually is like, it's a double layer. And if these are the oily parts, you know how oil is. It's kind of slippery. These 2 layers can slide on one another because this is slippery and this is slippery. And the cholesterol being added makes it even more slippery. We have integral proteins that go all the way through the membrane. They're also called transmembrane proteins because they go through the membrane. And we have peripheral proteins that are just on the surface, on either of the surfaces. And we have cytoskeleton proteins that are maintaining the shape of the membrane, maintaining its surface shape, its 3D look. So my question for you right here, looking at this, this is a picture, picture taken using microscopy to look at different organelles within a cell. What type of microscopy is it? What did they use here?
An electron?
They use an electron, but what type? Yeah, there's 2 different types. There's a transmission and there's a scanning.
Transmission.
They use transmission. Good job, Penny. They use transmission. Let's take 10 minutes and come back to right here. And we really, this will not take us as long as what we've done so far. So we'll take 10 minutes. So come back at 7:58. Come here, baby doll. Hey, Mom. Where are you, at Walmart or Aldi's? If you go down the aisle that's right next to where the paper towels are, the aisle that's perpendicular to that aisle that's next to where the pet stuff is. Yeah, if you go where you know where the pet stuff is, that the next aisle that's right next to that alley, the area, it's on the same, so where it's parallel to the pet stuff. And so if you look, the aisle where the paper towels is, and then there's that parallel aisle that's on the other side of that aisle is the pet stuff. Do you understand what I'm saying? You know where the paper towels are? Okay, walk down that aisle. Walk down that aisle so that you're headed towards the pet stuff. And then right in front of you is where they are. Yeah, right on the other side of that little center area. Yep, you'll see them there too. Do you see them? I've got to get off the phone because my classes, but if you don't find them, just leave them. Don't worry about it. Okay, love you, Mom. Bye-bye. Oh, awesome. Yep. Or it doesn't matter. Not the strips. It doesn't matter if they're which ones they are, Mom. Yes. Okay, Mom, I gotta get off the phone. Love you. Bye-bye. She's shopping right now, and she's wondering where in the store something is located. So I was trying to walk her through it. All right, coming back to this. Plasma membrane, this is just, the next two slides are a summary of those different organelles and what their function is. Plasma membrane prevents the escape of the cell contents. It just kind of encloses things. It regulates the exchange of materials between the cytoplasm, which is in the cell. Cyto, remember that means cell. Plasm meaning the materials within inside the cell. Extracellular fluid, extra meaning on the outside, cellular fluid. So this is the fluid outside of the cell. So the plasma membrane is regulating the exchange between what's in the cell and what's out of the cell. It's also involved in intercellular communication. What's on the outside of the cell can look like signs to other things and tell other things what's going on with that cell. It's kind of like having street signs and lets you know what's going on there. Microvilli. Micro meaning little, villi means like little finger-like structures. So microvilli mainly increase the surface area of a cell, which we'll talk about why that's so important. But it has a widespread sensory roles. Cilia means hair-like, and these move substances along a cell surface. Widespread sensory roles. Flagella are they look like little snakes or little tails, and they allow things to swim. And so sperm motility, the only animal cell that has a flagella are the sperm. Cytoplasm, so this is that area in the cell that includes the cytosol. Think of, so remember cyto, sol meaning just the liquid inside of the cell, and all the organelles. So the cytosol is that fluid portion of the cytoplasm in which many of the cell's chemical reactions are going to occur. So think of the cytoplasm as being the cytosol plus the organelles. And the cytosol is the cytoplasm, the cytoplasm minus the organelles. The cytoskeleton, remember this is the skeleton of the cell. It's got 3 kinds of protein filaments. And the 3 types of protein filaments are listed here. They maintain the cell shape and are involved in cell motility, movement, and movement of organelles. Cell movement and movement of organelles, allowing organelles to move within the cell. The microfilaments are protein filaments that are part of the cytoskeleton. They support microvilli. Remember, those are the little finger-like structures that are on the surface of a cell, and the plasma membrane. They're also involved in muscle contraction, endocytosis, remember that things coming into the cell, and cell division. The intermediate filaments are protein filaments that are part of the skeleton. They give shape and physically support the cell and anchor the cells to each other and to the extracellular material. They also compartmentalize cell contents. Microtubules are protein filaments that are part of the cytoskeleton. They help to form cilia and flagella, centrioles, and mitotic spindles. And they form the trackways. These are the roads that direct organelles and macromolecules to their destinations within the cell. The nucleus is the genetic control center of the cell. It contains the DNA. It also determines what genetic material is going to be made into proteins and what isn't at that time. It shelters the DNA. Chromatin is the assembly site for ribosomes. Remember, they're involved in protein synthesis. So wait, that's not right. That's an error. Chromatin is DNA. This right here is not right. This is DNA. And actually, let me do this. Switch in your PowerPoints, switch this and this. Because chromatin is long DNA strands within the nucleus with the associated proteins. The nucleolus is the assembly site of ribosomes. If I get a chance after class, I'll fix that and update the PowerPoint tonight. So the assembly site for ribosomes is the nucleolus. The chromatin is the long strands within the nucleus that are composed of DNA and proteins. The rough endoplasmic reticulum is the site for protein synthesis and manufacturing of cell membranes. The smooth endoplasmic reticulum is lipid synthesis, detoxification, and calcium storage. The ribosomes interpret the genetic code and synthesize proteins, which are also polypeptides. Polypeptides are also AKA proteins. The Golgi complex is involved in packaging products into vesicles, also involved in synthesizing carbohydrates. Secretory vesicles are involved in secreting things outside of the cell via exocytosis. Guys, I know that's a lot of information. Do the best you can and realize that we will spend all of chapter 3 in lecture also talking about it. Lysosomes, remember, they're involved in destruction of things. Peroxisomes also destroy things, and they use hydrogen peroxide to do it. The mitochondria are involved in ATP synthesis. ATP is the energy or the electricity for the cell. The centrioles form the mitotic spindle during cell division. They produce more ropes and to pull things certain ways. Unpaired centrioles are involved in forming cilia and flagella. If you remember from that picture of the centrioles from before, bear with me here, 16. The centrioles here are in pairs. When they're not in pairs, they're involved in forming the microvilli and the flagella. When they are in pairs, they are involved in forming the mitotic spindle or forming ropes that tug things around within the cell. The centrosome is the organizing center where the centrioles are located. And it's also involved in forming the microtubules of the cytoskeleton.
I'm sorry, Professor. Can you please repeat what you said about the centrioles? So
The centrioles are when they're paired, they're involved in forming the mitotic spindle, which we'll talk about why that's important in 1 second.
Okay.
When they're unpaired, they help form cilia and flagella.
Okay, thank you.
Welcome. Some basic, some terminology here. Passive transport was that idea that when there's 200 of us in one room and 4 of us in another room, we're going to move, we are going to move, and nobody's going to have to force us out of that room with that many people. We're all going to want to move anyway. That's passive transport. That's when something is passively moving and nothing, no energy is required to move it. It moves by its own energy. Active transport is when things are moving. Bear with me here. Active transport is when we're forcing things to move a certain way. Like maybe we're packing those 4 people into that room with 200 people. We make things move a certain way. To do that, we have to put energy into it. And our energy is adenosine triphosphate, or ATP. We also can use enzymes, which catalyze reactions that normally wouldn't occur passively. Remember, endocytosis is the moving of things into a cell. Exocytosis is moving things out of a cell. Phagocytosis is eating something. Phagio, like Xena when she jumped ahead there and said macrophage. Phage means to eat. And so phagocytosis is something being engulfed. Pinocytosis is little pinching or cell drinking. Things are just being pinched. Engulfing is going, ah. Pinocytosis is just little tiny pinches. That's the difference between these two. Engulfing is where you go, you grab it all, and you pull it in. Pino is just little tiny pinching, little tiny cell drinking. So phagocytosis means cell eating or inhaling. Pinocytosis meaning cell drinking. These little tiny pinches. Vocabulary describing chromatin. Remember, chromatin is DNA, the genetic code, DNA, with proteins. The chromatin is organized into chromosomes. Along the chromosomes are genes, meaning areas that are going to be the blueprints for a protein. What a protein is going to, it's going to be read to determine how a protein should be made. Chromatids are when we have 2 duplicate chromosomes, when the cell's getting ready to divide and we want to make sure that each cell is getting the identical DNA. We're gonna duplicate the chromosomes. Cell division is dividing one cell into another, or dividing one cell into 2. Gametes are your sex cells that contain only half of your genetic DNA. Because you'll find that special significant other and combine one of your sex cells with one of their sex cells to give the embryo its full genetic content. So somatic cells are your everyday cells. Your cheek cells are somatic cells. They contain all of your DNA. Every one of your cells, except for your gametes, which are your sex cells, contain all of your DNA. And when they contain all of your DNA, they're called somatic cells. What that also means is that you've received, for each of your chromosomes, you have two. You have one that came from your mom and one that came from your dad. Di, just like bi, when we are looking at binocular, di means two. So this is when you have two chromosomes for each designated chromosome. You have a pair. Gametes are only haploid. You think haploid meaning half of the genetic material is there. Now, I will tell you this word gametogenesis. Genesis is by far my most favorite base word. Where in the world have you ever heard the term Genesis? Just Genesis by itself, Genesis.
The Bible, or it means like the beginning.
Yeah. What? Okay, it means the Bible. So it's the first chapter of the Bible, and it means the beginning, or what? What else can it mean, Christina?
Like birth, maybe.
Maybe the first. What happens in that first part of the Bible?
Creation.
Creation. Anytime that you see the word Genesis on the end of anything, it means the creation of something, the building up or synthesizing of something. Zena, you have a cute kid there. Gametogenesis is, you're welcome. Gametogenesis is the synthesis of gametes. We will see this word a lot, especially Genesis. And just remember, it's all whatever's in front of it is what it's synthesizing. Oh, one more word, cytokinesis. Remember what cyto means. What does cyto mean? Cell. Kinesis means to cut. So this is divide, and it's specifically the cytoplasm. So this is dividing the cytoplasm in half. Cytokinesis is dividing the cytoplasm in half. And we're going to talk about the cell cycle. The last part of this is talking about the cell cycle, or somatic cell division. So this is specifically somatic cell division, meaning we're not looking at the formation of gametes. We're looking at dividing cells, and the daughter cells, the cells being produced, have the same amount of DNA as the parent cells did. So what we have to do is we have to duplicate all of the DNA so that it can all be, so we can split it evenly into both cells. The first part of this is called what's called interphase, interphase. And what that means is it's involved in the building up of the parent cell that's going to divide, and building that cell completely up, duplicating all of the organelles, duplicating all of the DNA, and then making sure that all the cell is big enough and that the DNA is all duplicated before we divide it. So the major part of the cell cycle and the most amount of time that this cell spends in the cell cycle is going to be spent in interphase. A lot of times we have cells that are going to leave the cell cycle and they're not going to divide. We'll talk about neurons in a little while. They don't divide. Overall, as a general rule, once you are born and once your brain is completely formed, you don't generate more neurons. What they do is they enter this phase called G0, where they just don't, they're not going to divide again. Cells can come back out of G0 and enter the cell cycle again, but if they're in G0, they're in a point where they don't proceed through the cycle. Every once in a while, and I know of a really neat experience where neurons can recover. But as a general rule, they're one of those cells that go into this G0 phase and they don't come back. Muscle cells are another one. And we'll talk about that later, towards the end of the semester, that muscle cells don't divide. So interphase is the major part. We duplicate all of the organelles, we duplicate all of the chromosomes. And then we start dividing up the DNA. That part where we're going to divide all the duplicated DNA is called mitosis, which actually means the division of the nucleus, because remember, the nucleus is where the DNA is contained. There's several stages to mitosis. And you can see that interphase is technically not part of mitosis. So we have this prophase, this metaphase, anaphase, and telophase cytokinesis. It's going to occur after that right here. The mnemonic, and I have a great mnemonic for remembering these phases. It's called ITMAP. Oh, gotta put it in the right word. ITMAP. And what that means is that the phases from interphase all the way through mitosis are interphase, prophase, metaphase, anaphase, and telophase. And that's the way that I can remember the order of these phases, is ITMAP. And if I just want to remember the phases of mitosis, it's just PMAP. So we start in prophase, where the centrioles, remember, they are the ones generating microtubules or the mitotic spindle. The mitotic spindle is going to reach out to the duplicated chromosomes. and hold on to them. This is occurring during prophase. We're generating this mitotic spindle that's reaching out to these duplicated chromosomes. And what it's doing is it's tugging them and pulling them to the center of the cell. Tugging them and pulling them to the center of the cell. And then half is going to go one direction and half is going to go the other direction. This is anaphase. So telophase is when we separate into two separate cells. Cytokinesis when we sever them. So we duplicated the DNA. Then we have to figure out how are we going to separate it into two separate cells. So that's where the mitotic spindle is. We have to make sure it's an even half, going from one half into one cell, half into another cell. To do that, we're going to have them all meet up in the middle. I think of meta as meaning in the middle. They're meeting in the middle. Anaphase is when we're separating them, we're taking half of it one way and half of it another way. I think of ana as meaning a dividing point. We're dividing half one direction, half the other direction. Telophase, I think of telo as meaning a tail end. So this is the end before cytokinesis. I will tell you that if this was a general biology class, we'd spend a week on this. So you're getting a lot really fast. So we've got this cell right here. Here's those centrioles that are generating out these mitotic spindles or spindle fibers to connect up with these chromosomes. And they're pushing and pulling them so that they all line up in the middle. So early prophase, prometaphase, metaphase, anaphase, and telophase.
No.
In early prophase, we've just gone through interphase. The centrosomes have duplicated. So the centrosomes are what produce where the centrioles are located. And they're the ones that are going to develop the mitotic spindle. The chromatin is condensing up so we get thick chromosomes. We need the thick chromosomes. We want the DNA to compact up here. And the nuclear envelope is fragmenting. Why? I'm going to ask a really quick question here. Why do you think the DNA is compacting? Why do you think that's important that it compacts up here? It's very loose in interphase. Why is it all compacted here? Why do we need it to get all compacted?
To ensure that it evenly divides.
What and how is compacting it up going to help it evenly divide?
make
to
sure
divide it equally?
nothing's forgotten?
Say it again.
Is it to make sure that all the parts are there, that like nothing is missing?
That's exactly right. You guys are smarter than some of my general biology students. Because when I ask them that, they don't really know what to tell me. And I ask that about every time. So that's exactly right. It's a lot easier to separate thicker strands than a bunch of thin, diffuse stuff that's all tangled up. If you know of genetic disorders where people are missing parts of chromosomes, this is maybe where it's occurring. I have a niece who she has what's called Williams syndrome. And what that means is that she's missing a major part of chromosome number 7. And whether it's occurring here or it's already occurred, I don't know. But they have to compact if we're going to be able to separate them evenly. So prophase, the nucleolus disappears. Remember, the nucleolus is what contains the ribosomes in the nucleus. And the duplicated chromosomes are now visible because they're compacted up. The centrosomes then begin moving apart. Remember, they're the ones that contain the centrioles. They start moving apart and the spindle is being formed. And it's reaching out to the duplicated chromosomes that are compacted up. In prometaphase, each of the chromosomes are being attached to a spindle and they're pushing them towards the middle. Pushing and pulling them towards the middle. Metaphase, they're duplicated, the chromosomes are aligned at the center. And now the ropes start to shorten to push half of the DNA one direction, half the other direction. Anaphase, we're pulling them apart. So the sister chromatids are parting and they're becoming daughter chromosomes that move toward the spindle poles where the centrioles are. Think of poles as like the poles of the earth. They're moving them towards each end. In telophase, the daughter cells are forming and the nuclear envelopes are coming back to enclose the DNA. And the chromosomes are decompacting, they're becoming diffuse again. This is just another summary slide explaining this process. I'm not expecting, like I told you, in a general biology class, we'd be spending a whole week on this, or at least a good 2 hours. I'm not expecting you to completely understand it. Get the general idea. When you're looking under the microscope, you can see cells in various different phases. Some in metaphase, some in anaphase, some in prophase. If you're looking here, could you distinguish them? We're not going to worry so much about that right in this class. What are the functions of mitosis? It allows for embryonic development. It allows for the little Xena's little minion to grow up. The healing of wounds. You get injured, it allows you to heal. And to replace cells that tend to wear out. Genetic expression involves changing the DNA to an intermediate of RNA and then changing RNA into protein. The transcription is coding the DNA, taking the DNA code and putting it into an RNA code. Translation is taking the RNA code and putting it into a protein code. Be familiar with the terms. Spend some time looking at this and being familiar with the terms, but realize that you don't need to know it really in depth. We'll spend time in chapter 3 talking about this. We won't talk about mitosis, but we will spend some time talking about this. Do you have questions for me? Probably a lot. It's a lot of material.
This last part on gene expression, is
Say
that
it, say it one more time. I'm sorry, start over. Say it one more time.
This last part on gene expression diagram, is that the book or the lab book?
I don't, let me look. This figure is actually from another book that I have. But let me look really quick. It may be that it's not even there. What I want you to do, some of this is not coming exactly out of your manual. Recognize that some of this material, like when we're talking about the electron microscope, this is the time that we have to talk about it because it's related to the fact that it's also their microscopy. But it may not be part of the lab. I just know that it's going to build onto the subject. Does that make sense? When you're doing the questions, when you're looking at the questions, I'm not going to ask you about transcription and translation on your lab quests. But they will be coming up in lecture again. And sometimes lab is mainly to reinforce and allow you to repeat and repeatedly hear different things. Does that make sense? I brought it up in here because we talked about DNA. And we talked about protein synthesis and that this is what some of the organelles do. That's the reason I bring it up. I don't think this is in there, but if you remember, the ribosomes are in there. And the polypeptides are in the book. This polypeptide is a protein. Does that help? Kind of. Or just make you frustrated? All right, she's not responding. She's probably frustrated. You guys, this is okay. This is okay, good. This is the end of our class. Zena, would you, I think she just went away, but Zena, would you, if you're still here, would you stay for 1 second and let me talk to you?
Sure.
Okay. Guys, have a wonderful night, all right?
Thank you. You
Thank you. You too.
Thank you.
Hi.
Thank you.
Welcome.
Thank you. See you.
See you. Zena, the reason, and this doesn't need to be really private, are you taking this class for credit and that class for credit?
Yes.
Okay. That's fine by me. Are you sure? Have you talked with your advisor? Are they going to allow you to get credit for both classes, taking them at the same time?
I was going to transfer to be a college after I took it, and they said yes, as long as I take those, because I had taken both classes before, but they're about to expire now, so I won't be able to get into their nursing program without taking this 2 courses for June. So that's why I'm in a time crunch. So I had to take both of them, but because I took them before, they just wanted me to either take a refresher or take these classes, and I chose to take the classes. That way, you know, I can like, it will refresh my memory, and it will help me.
All right. I saw with your grades, I saw that you didn't have the homework for this class. So I thought, is she auditing it? Or let me, bear with me. I'm going to stop sharing and I'm going to stop recording.
Okay.